The history of immunology is a relatively new area of science. I mean, there's some ancient history where people tried to come up with ways of treating disease, and they recognized symptoms of the immune response in that in the fact that some people, once they had the disease and got over it, were completely immune, and that those people were usually designated to take care of the sick that had a disease. George Washington used a form of immunization where he cut the arm of some of his soldiers and inoculated them with the disease that was attenuated so that they would not get the disease in a more virulent form. Um, the Chinese and the Turks did a process called variolation, in which they tried to prevent smallpox by taking the pus from milder cases of the disease. They would pop the pustules and collect the pus, dry it, and then have people inhale or snort the dried, ground up pus. And then hopefully it would give them a milder case of the disease. Well, there was a British lady, Lady Montagu, who lived in Turkey for a while as it was part of the British Empire. Um, Lady Montagu had that same technique done on her children, and when she moved back to the British Isles, she brought the idea of the technique with her. And it went on for a few years until one of the royal family actually tried it and got sick and died from the disease. So one of the princes died and then it was outlawed. So now, then we're going to just very quickly go over some of these because I have individual videos for Edward Jenner and Louis Pasteur, um, monoclonal antibodies. I don't have one for Metchnikoff, but I'm going to go through this really quickly. But you do need to go back and watch the videos for Edward Jenner and Louis Pasteur. Those are two of the big names when you come to immunology. All right, so in 1798, this is after variolation was tried in England. Edward Jenner was hanging out with milkmaids, and the reason was is they were a lot prettier. They always had smooth skin. They never had scars that were caused by smallpox. So he was trying to figure out why. Well, basically, milkmaids were slaves. They were usually girls that were sold to the dairy farmers when they were very young, and their job was to take care of the cows. They would milk the cows in the morning, and in the afternoon they would carry water and food for the cows and that sort of thing. But they were also known to be some of the prettiest women in any community. So it was actually an older milkmaid that told him because they got the cowpox on their hands that they never got the smallpox, and that gave him the idea. So he was able to take a young boy from a neighboring village, and this is before hypodermic needles were made. So he sharpened a piece of wire in a flame, as sharp and thin as he could get it, and he had dried the pus from cowpox pustules, and he dried it, and he injected it under the skin by repeatedly touching the dried pustules and sticking it in the skin of this little boy over and over again, multiple times. And then a few weeks later, he challenged that same boy with the strongest smallpox pustules that he could find. Now, I don't know where the kid's mom was, but that's how he came up with the vaccine. Then in 1881, Louis Pasteur actually came up with several different things. He was able to vaccinate sheep and other farm animals against anthrax, which was a major deal because anthrax was killing a lot of the farm animals in areas all around Europe, and it was affecting the food supply because you couldn't eat an animal that died from anthrax. Louis Pasteur actually came up with the term for vaccine. Vaccine literally means encowment, to turn into a cow. And I'll talk more about that in the individual videos that are out there. Then in 1883, Metchnikoff, who was a weird scientist, he was a little on the suicidal side. He was very arrogant. If you ever get a chance to read a book called Microbe Hunters, it's really good. And it's kind of the gossip of how these people worked. And he found that white blood cells can phagocytize. I think he took water fleas and crushed the water fleas, and he was able to show that white blood cells from mice or other animals were able to engulf. It may have been phagocytes from the water fleas that were able to engulf bacteria, and he was able to show phagocytosis. Then in 1913, anaphylaxis was, I skipped some. I skipped 1901, von Behring and Kizziato, and they both demonstrated that serum could transfer immunity. So if you take serum from someone who survived the disease and give just the serum, not the whole cells, to another person, that it could transfer some passive immunity in that context. Anaphylaxis, which was in 1913, two French researchers thought they were going to come up with a vaccine against Portuguese man of war. These are found in the oceans, especially in the south of France, and some people get stung and it just causes welts and they hurt, but some people would go into anaphylaxis and die from it. So they wanted to inject dogs and then come back and inject them again with the nematocysts, the stinger parts of the tentacles from these Portuguese men of war. And what they found out was that anaphylaxis was caused by too strong of an immune response. In 1930, human blood groups, they were able to start typing and cross-matching, and then that kind of started the whole revolution of being able to use blood from one donor to save the life of someone who's lost quite a bit of blood. It wasn't until 1957 that antihistamines were found. In 1977, the last known naturally acquired smallpox case occurred. In 1980, they discovered major histocompatibility complexes. So you see, really, a lot of this stuff didn't start up until the late 1900s. So this is a very young field of study. And I do predict that we will be able to use immunology or immunological techniques to cure and treat a lot of diseases, including many types of cancer. So there's breakthroughs almost daily in this field. 1984, monoclonal antibodies, which are a major breakthrough, and you will learn all you want to know about how to make monoclonal antibodies and how you can use them in the course of this class. So there's a little bit more here. 1798, Edward Jenner's the one that I've already talked about. Since I have separate videos based on these two individuals, I won't go into much detail here. Just suffice it to say that Edward Jenner and Louis Pasteur are very influential in the beginning idea that the immune system was working. Excuse me. The immunizations and vaccines and things like that have really saved a lot of lives. In industrialized nations, measles, mumps, whooping cough, tetanus, polio, diphtheria are extremely rare or almost non-existent. We are having a reoccurrence of certain diseases like measles are being brought in by immigrants from other countries. Mumps and whooping cough are coming back. A lot of that's due to the anti-vaxxer movement where people think that the vaccines are just horrible. I will jump up and down and scream and yell and call you every name in the book because vaccines do work and we've got tons of evidence to support it. I'm sorry, I just hate the fact that somebody can spend 30 minutes searching the internet and think they know more than someone who spent 20 or 30 years studying the process of vaccinations. Okay, I'll get off that soapbox. We want to be able to prevent the spread of disease, and it's also helped us to lengthen the lifespan of a lot of people. So vaccines really are important. Um, Menchikoff already mentioned, basically, he worked on phagocytosis, but the ones that I didn't talk about much were von Behring and Kitsia Sato. They demonstrated that serum, non-cellular components of the blood, so if you take the blood and you let it separate into the clear liquid that's on the top, that's serum, and it contains something in there that we now know of as antibodies and possibly even complement cascade proteins. That you can take that serum from a survivor of a disease and put it into someone who's naive or never been challenged before, and it will help to transfer some immunity to non immunized animals. It doesn't give long term immunity, but what it does is it neutralizes the virus or the bacterium um, in the short term, which will allow the patient who has the disease to survive.